everyone. Thank you so much for joining us. Before we kick this off, I do want to point out to the audience that we welcome questions. Please post them in the Q&A section at the bottom of the screen, and we will get to those questions at the end of today's fireside chat, which will be led by Steven Saltzstein, Force Family Office's CEO. And Steven, I'm going to turn this over to you. Thank you.
Thank you, Callie, and thank you everyone for joining us today. 30 to 50% of patients with rheumatoid arthritis, lupus, type 1 diabetes, other common autoimmune diseases do not respond to standard of care. Obviously that is a issue that needs to be solved and corrected, and that is exactly what Autoimmunity BioSolutions is doing. So it is my great pleasure to welcome Eugene Williams. Eugene, thank you so much for being with me.
Hey, Stephen, thanks for having me, and thanks to everyone who's joining today or maybe catching up on recording. Really appreciate your interest in ABS.
So just to begin with, for those that don't know, can you explain what is autoimmune disease and what causes it, and how prevalent is it?
Yes, an important question. So there are about 25 million people in the US who have autoimmune disease. And the right way to think about autoimmune disease is friendly fire from your immune system. So what happens is when you're very young, there's something called thymic education that tries to teach your immune system the difference between self and non-self. It's really pretty amazing when you think about how your immune system can tell the difference between all the proteins in your body and something that's a foreign protein like a virus. But it works really well. However, when you get older at times, sometimes things break down, and that causes an autoimmune disease where your immune system mistakes you for a foreign antigen like a virus. Now, one of the ways that can happen is something called molecular mimicry. Autoimmune disease happens often in people who are infected with Epstein-Barr virus. Now, the way that works is there's something about that virus that looks a lot like your tissue. So when the immune system responds to that virus, it accidentally starts mistaking your tissue for a foreign antigen and it attacks it. Many people who have MS were exposed at one point to Epstein-Barr virus. So that's one of the processes by which your immune system can start to make mistakes. And an autoimmune disease is essentially friendly fire from your immune system.
Oh, that's great. That's a great explanation. Thank you. So tell me about the company. Tell me about ABS, as we call it. What's the history behind ABS?
Yeah, so ABS has a 20-year history. It's founded by a couple of geneticists, and to focus very much on what you started talking about at the beginning, Steve, which is what causes lack of response or greater severity in autoimmune disease. So you remember about 25, 30 years ago, the human genome was cracked. And at that point, lots of genetic information was made available to many, many scientists. Our two founders are geneticists. So they started looking at associations between genetics and disease. And in autoimmune disease, they discovered something 20 years ago, which was correlated with much more severe disease in MS, much greater risk of progression. It's a genetic variation called a SNP. It's a single nucleotide polymorphism. Everybody has some of these somehow. Sometimes they're meaningless. Other times they lead to differences because gene is a recipe for making a protein. Patients with this SNP made 3 times as much of a protein that's an important component of your immune system. That protein is called soluble IL-7 receptor. So to boil it down, our scientists discovered a correlation, an association between a genetic situation that affects 53% of the population and much more severe autoimmune disease that doesn't respond well to therapy. So in the subsequent 20 years, as often happens, the scientific community picked up on these early findings and discovered in one autoimmune disease after another, this biology, genetically elevated protein, was associated with a poor response to therapy or more severe disease. There have been no exceptions to the best of our knowledge. So in one autoimmune disease after another, this biology that our science team discovered was associated with a problem that needs to be fixed. Now, one of the important things that happens then in thinking about these genetics is it a cause or a consequence? So at the NIH about 12 years ago, very, very respected scientist called Krista McHale did a study with animals where she showed that she could make disease worse by adding this protein. So that was a very, very strong indicator of causality. Put it all together, And about 4 or 5 years ago, our founding scientists, who were both geneticists, decided to start the company. About a year and a half ago, we ended up being a real biotech company, if you will, by raising our first investor round. But now we're working hard to solve the problem you started at the beginning. And by the way, one of our founding geneticists, Mariano Garcia Blanco, was just inducted to the National Academy of Sciences. So we're very, very pleased for him.
Biotech M&A has been really hot, and autoimmune especially. So, why do you think that is? Why is autoimmune is such a wanted asset right now for, let's say, large pharma?
Yeah, I think, first of all, it's a 25 million population where there's a 30 to 50% non-response. Huge TAM, lots of unmet need. And, if you look at, you talk to physicians in the autoimmune disease area, I think one of the key issues they point to is this 30 to 50% non-response. What do I do for my patients when none of the therapies I have already are working effectively? So what pharma has been buying are novel approaches to a different way of doing immunosuppression. We'll talk about this probably a little bit more, but our approach is different. We're looking at immunocorrection, not immunosuppression, but the vast majority of treatment in autoimmune disease has been just a different way of suppressing the immune system enough to mitigate disease, but not so much to compromise the patient. Now, because there's so much unmet need, pharma recognizes we need a lot more novel therapies, which is why the M&A area has been very hot. Our plan is to target an M&A exit, but we're trying to do it in a way where we're going to try to position our company as one which is a novel, broad-based approach that can make all those other therapies work a lot better for a large portion of the population.
So I want to put a pin in immunocorrection because it really is personalized medicine. I want to come back to that, but just can you just tell us what are the current treatments? What are the current standard of care treatments for autoimmune disease? How do they work?
All of them work through immunosuppression. This actually started 40 years ago with something called cyclosporin, which you may know was the product that enabled the transplant market. I worked on this actually 40 years ago. After it was done in transplant, we looked at it in a bunch of different autoimmune diseases. So the idea there is if your immune system is attacking your body, if you dial down the immune system a bit, then you can stop that attack on your body. But the key there is you need to strike a balance. Now, in the last 40 years, there have been a whole bunch of different types of immunosuppressives that have been developed. The products like Humira and Remicade are called anti-TNF antagonists. There's IL-6, there's IL-1, there's IL-13, IL-17, IL-23, CD20, CD52. You get the picture. Lots of different mechanisms that are involved in your immune system in a healthy immune response, where the therapeutic goal is to try to strike a balance. That's a tough thing to do. Try to dial down the immune system enough to mitigate disease, but not so much that the patient's subjected to horrible risks of adverse events from infections and so on. Now, there's an inherent problem in that. And this 30 to 50% non-response you talked about means that patients are so heterogeneous that for 30 to 50% of the population, that balance doesn't provide adequate relief. So what we're trying to do is coming at it from a different angle. We're identifying what's the reason for many of those patients not responding to standards of care. Let's fix that reason. That's what we call immunocorrection. And a large portion of those patients who don't respond, it appears from the literature and work that we'll do, don't respond because they have genetically elevated soluble IL-7 receptor, which is the problem we're trying to solve with our antibody therapy.
I know you're not going to like this question, but I mean, it kind of, because you're doing immunocorrection, it kind of, for me, has a CRISPR feel. A lot of our family offices out there know CRISPR. They know the mechanism of action. Can you just give us a compare and contrast?
Yeah, so it's a very interesting question. It's a very good question because there are actually two different therapeutic approaches that we could take on which we have IP. Now, we've focused on our antibody therapy because antibodies are very, very well known. They can be conveniently delivered, sub-Q injections. Most of the problems in getting them to work the way you want have been solved multiple times. So that's why we're focused there. But you talk CRISPR, it's really gene editing. That's the other approach in which we have IP. There's something called ASOs or antisense oligonucleotides. What that is essentially splicing a correction into a gene that's making what you don't want it to make. So what we talked about is this genetic association. What our scientists found is 53% of the population have this SNP, which, if you want to get really technical, skips exon 6. But what it does is a gene's a recipe for making a protein. That recipe makes 3 times as much of the protein as is normal and as you want. So for patients who have that SNP, that genetic condition, they make too much of the protein. Now, the other way you can fix that, similar to CRISPR, is by splicing that gene. In other words, change the recipe so the patient makes less of the protein. Now, we have IP on that. I think pharma likes the fact that we have that other approach, that complementary approach, that's worked really well in non-human primate models. But again, the main program we're going to take forward is our antibody program, which achieves the same goal a different way. It says, okay, I measure a patient who has too much protein, my antibody reduces it to the normal level. You can achieve that also by changing the recipe by using an antisense oligonucleotide, similar to CRISPR. But the more convenient, practical way for us to do it is focus on the antibody. But then what pharma might buy in an M&A transaction is the fact that we have IP on both approaches.
Right. And would you say, I mean, how strong is the IP portfolio? I mean, would you say that you have this approach pretty much locked up?
I believe it's very strong. Again, we have consultations with, really strong intellectual property attorneys. They do, evaluations to see whether we're first to file, whether we have freedom to operate. To the best of our knowledge, we're the only ones who have focused on selectively targeting this protein called soluble IL-7 receptor. There's another form of it called the membrane-bound receptor. There's, the protein it binds to called IL-7, but we're the only ones, to the best of our knowledge, who filed IP targeting selectively the soluble receptor, and that's what's necessary for this selective immunocorrection. What we want to correct is patients who have too much of the soluble receptor, so you want to leave everything else alone and only correct that. To the best of our knowledge, we have the only IP to that effect.
And so do you look at this as a combination therapy or?
Absolutely right. Yeah.
So standalone, it's combination?
It is combination. I think, one way of thinking about this as well is, I know a lot of your audience is very sophisticated in healthcare and biotech, is we're borrowing a lot of things that have been proven in oncology. So in oncology, we talked about precision medicine or personalized medicine. That's where it first took off. And the idea is treat the patient with the therapy that's most likely to work for them. And there are biomarkers or genetics or other things that can give you that information. So that's part of what we're borrowing. But then the other thing we're borrowing from oncology is this concept of a mechanism of resistance. So a lot of what happens in oncology treatment is tumors become resistant. There are biologic mechanisms by which they resist treatment. Well, we believe the right way of thinking about what our scientists discovered 20 years ago is genetically elevated soluble IL-7 receptor is a broad-based primary mechanism of resistance to immunosuppressive therapy. So if you have too much of this protein that resists, your immune system responds too strongly to your own tissue, and therefore it resists adequate treatment from the immunosuppressives. So the way in oncology you treat, a resistant patient is combination therapy. You combine something that addresses the resistance with chemotherapy that might work if the patient didn't have the resistance, and that's exactly what we're doing in autoimmune disease. I think we're in the vanguard of a bunch of areas like precision medicine, like combination therapy, that I think have been really well established, work really well in oncology, and moving into autoimmune disease, I think we're in the lead on that.
Now, what's interesting is you guys have the biomarkers. So basically you have the diagnostic as well theoretically as the cure.
Exactly right. Yeah, and that's very important for our strategy. And it also gives us some great opportunities in addition to just the therapeutic opportunity. So in order to treat patients in a precision or a personalized way, you need to measure whether they have the problem you're trying to solve. So again, what we're trying to solve is genetically elevated soluble IL-7 receptor. So we have assays that measure that SNP. 53% of the population has that SNP that gives you too much of the protein. We can measure that. And we have a CLIA certified process in a lab in New Haven that can do that per all the appropriate standards. The other thing we want to measure is the level of the protein. We also have an assay in that same lab that can measure that level of protein. It's called an ELISA assay. So that's key to our therapeutic strategy. When we want to treat patients in the clinic, what we'll do is we'll measure to make sure that they have the problem we're trying to solve, because our treatment benefit, the result of our trial is going to be most compelling if all the patients in that trial have the problem that we're trying to solve. The other opportunity it creates for us is as we go out to pharma and we're cultivating relationships and awareness that ultimately hopefully will lead to an M&A exit. There are 20 large pharma companies who have 3 billion plus revenue in the autoimmune space, and we're trying to have conversations with all of them. We're making them aware that for their own portfolio, for products in development that have variable response, maybe on a road to failure, we may be able to help them identify a subpopulation where their therapy works really well. And then in the long run, if they buy our therapy, they can work for the other half of the population. So we may have some near-term diagnostic, either partnership or revenue opportunities in advance of when we finish our phase 2 study a few years down the road, and hopefully leading to an M&A exit. So you're right, we needed both therapies and diagnostics to do or carry out our strategy, and we have both.
So it's interesting. I feel like there's a lot of value at the company. Like one question about the diagnostic is theoretically, could it be a 510(k) approval or no on a standalone basis or?
You're in the right direction. Probably not 510(k) because I don't think there's an adequate predicate, which is what that needs, but a PMA approval, which is basically a diagnostic approval. That's absolutely the path we would want to take.
And it's interesting. So if I was an investor in the company, I mean, the one thing I would love is you have a lot of shots on goal that can unlock value because you have the diagnostic. Yeah, it's interesting. You have the diagnostic and, you even have beyond autoimmune.
Absolutely right. And I think that I was at Genzyme for many years where I was the general manager of the autoimmune disease business unit. And part of how Genzyme became a very successful company with therapeutics in rare diseases. But the way Genzyme paid the bills at the beginning was with a successful diagnostics business.
Right. Huh, that's great. So talk to me about the company. Talk to me about ABS in the sense that, like, what can we expect near term? And then what are the ultimate business goals?
Yeah, let me work backwards if I can from the ultimate business goals, because that tells you what we're going to do in the near term to get there. I'm a huge fan of starting with the end in mind. If you look at biotech, you never know for sure until you get into the clinic whether the science is going to work. But what you know for sure is that you have to be very clever in how you translate that science into the clinic so that you maximize the benefit if the science works. What I've seen so many times, and I hate to see, is good science, trials that didn't show it, or trials that were so mixed in their results that the early investors don't get the value they deserve, or patients don't get the benefit they deserve. So we're certainly looking for an M&A exit. And now how do we maximize, the value we get there? There's two components. One is what's the actual exit value at the point of an M&A exit, which is the end of a phase 2 study in RA. And then the second bucket is how do we optimize the odds of success and the cost and time frame to get there? So optimizing odds is increasing the odds, lowering cost and time frame. So let me start with the first bucket first. It has two components. One is how do you maximize the value of an exit? First is competition. So I talked about these 20 large players. We're in active discussions right now with a dozen of them and trying to get to work with more, hopefully trying to get some of them to invest in our Series A round next year, hopefully collaborate with some of them on biosample studies that we're going to do in advance of our clinical trial, and find lots of ways to work with them so that they really understand the potential value. We create that fear of missing out. The other thing that gives you a very strong M&A exit value is what's the risk-adjusted NPV when you exit. So let me give you some numbers.
Yeah.
So the total addressable market we talked about globally is probably 25 million, 60 million global, 25 million with the problem we're trying to solve. So let's be conservative and say only 5 million get on our treatment at peak sales, 20% penetration. Now, because how do you get a big penetration? Low price. Most antibodies are $30,000, $40,000, $50,000 a year. What if we price at $5,000 a year? That's a $25 billion peak sales.
Yeah.
Now that's in the range of the biggest products in autoimmune disease. That's a $100 billion NPV at launch. That's a $30 to $40 billion risk-adjusted NPV when we finish phase 2. Now you don't get all of that, obviously, but you can get a high portion of that, several $billion in an exit, if you have competition and you build a really strong case for that TAM. So part of how we're going to build a really strong case for that TAM is not only to do our study in RA, but do biosamples in multiple other autoimmune indications to expand the understanding and hopefully involve pharma in this so they become aware of the broad total addressable market in the process. Expand the understanding that for a large portion of patients with almost every autoimmune disease, non-response is driven by the problem that we're trying to solve. So that substantiates a large TAM, gives us a large risk-adjusted NPV. Now, the other side of the equation then is how do we avoid dilution by being very capital efficient and accelerate the time frame and raise the odds of success? So we're trying to do a few clever things there, I hope. One is in optimizing the odds of success. In these biosample studies, we're going to look at patients with RA, and we're going to use our biomarkers to figure out how do you define the population that doesn't respond well to Humira? How do you define a subpopulation that's 30 or 40%, where the difference in treatment response to Humira is like threefold? If you do that, think about it then, we've isolated the biology. So the only difference between this level of response and a threefold level of response is the protein that we're going to reduce, which means the odds of success in our trial, never 100% in biotech, but they're higher than they might be if the only data we had in advance is an animal model. So we've isolated the biology, raising the odds of success, raising the odds that we might get a result that says twice as good as Humira alone. Then the other things from a timing point of view, two things that cost a lot of money and take a lot of time in clinical trials are enrolling investigative sites and then patient enrollment. And the two feed back on each other. If patient enrollment's slow, then you need more sites. It gets you in this cost and time doom spiral. We're addressing both of them. What we're going to do to create these biosample data is involve a lot of investigators in advance of the clinical trial, and we'll pick the most motivated, best ones who have seen that the problem we're trying to solve is in fact the problem affecting their patient population. So we'll really streamline getting a lot of investigators up and running. The other thing is because it's combination therapy, think of the patient enrollment proposition for a patient with RA. Unlike the usual trial, where they're taking a risk of getting a placebo, here we're saying to a patient who has RA, they failed methotrexate, the first-line therapy, they're in pain, they're anxious. We say to them, you're going to get Humira no matter what. Now you have genetics that suggest you may not respond as well to Humira as some people. So for the 12 weeks of the trial, 50-50 chance you're going to get something that may make you respond better, ABS therapy. And if you don't get it in the 12 weeks of the trial, you'll get it in week 13 and later in the open label extension. So we expect enrollment, that to be a very attractive enrollment proposition. So we have a lot of investigators lined up. We enroll rapidly. We think we can really economize on the cost and time frame of getting to that exit point.
There's a bunch of audience questions, and I want to get to them, but I want to ask you, I just want you, if you don't mind, to really suss out your pipeline. I just want to make sure people understand, what phase you're in, what you're going to go for, what diseases you're going to focus on, and when.
Yeah, absolutely. So you did ask the question about the near term. So what we have now is antibody therapy and ASO therapy, as I described before. The antibody therapy is still a mouse therapy. So a lead and a backup have been identified. So what we're going to do going forward then are there are ways of what's called humanizing that antibody so that it can then be used in a clinical trial. And after that, what you do is once you have a humanized antibodies, you create a manufacturing process. So the typical way that's done for antibodies is you take the DNA of that antibody and you put it into a cell line. It's called CHO cells, Chinese hamster ovary. Don't ask me why. It's just the main production line for antibodies. And then you put those in bioreactors and they make enough material that you can use in a clinical trial. So that process takes about 12 to 18 months total. And once we get there, we can file for an IND because we'll do safety work called GLP toxicology work in parallel. The first study we'll do after we get an IND will probably be in Australia, where there's a tremendous tax rate savings. It costs half as much as the US. We'll be treating healthy normal volunteers who have the SNP. So we know they have too much of the protein. So in healthy young 25 or 30-year-olds, we can show that we can knock down the protein the amount we want to safely, and it'll give us a lot of information for that phase 2 study. Once all that work is done, the final development of the antibody, the manufacturing, phase 1 study, then we do the phase 2 study. And again, depending on time frame of capital availability and other factors, That could be in the 3 to 4 year time frame till we get the data that could lead to an M&A exit.
But let me ask you, so maybe I misunderstood, but so what about lupus?
Yes, that is the same antibody, the same product could work in all of those different indications. So what we'll probably
Okay.
do is instead of actually running trials in all those indications, that we may run 2 trials, one in RA, one in lupus, depending on what the interests are of the investors in the Series A round. 2 scenarios. One is we run RA alone. The other is we run RA and lupus. But for all of these other autoimmune diseases, instead of just running a trial with our therapy, we'll run an outcome study showing that genetically elevated protein causes non-response or leads to a great deal of non-response. So that'll be basically making the case that in all these other autoimmune diseases, if you were to do what we did in RA or in lupus, it should work the same way. If you take a patient with high protein who doesn't respond and you knock down the protein to the level where patients do respond, you'll probably get a great clinical response. So we're going to try to build a very strong, I'll call it circumstantial case for many other autoimmune indications. We'll definitely do a study with our antibody in RA. We might do a study in lupus. That's to be determined based on how much capital and what the appetite is for the investors in our Series A round. If, for example, we had a large pharma invest in the Series A round, which we're trying to get done, who put a high priority on lupus, and they wanted us to do a trial in lupus, and we raised adequate capital for it, then absolutely we would do it.
So when you look at the company, 5 years from now,
Yeah.
what is the complexion of the company? What does it look like? I'm guessing it's bought by then, but I'm just curious.
That's precisely what I'm expecting as well, or guessing, or hoping for, shooting for. We'd be acquired by that time. A large pharma company would be in a pivotal trial, probably in RA, maybe in a second indication like lupus as well. And our biomarkers would be being utilized, hopefully as a PMA-approved diagnostic, helping physicians in those autoimmune diseases identify likely responders and non-responders. So in other words, building the market, helping people identify the patients who could benefit from our therapy because they have genetically elevated protein. That's why they're not responding to current standards of care. So that market development process, which Big Pharma always wants to do prior to launch, would be made, I think, dramatically more powerful in our circumstance, because we could go to every physician, offer them a diagnostic and say, if patient's not responding, this may give you the answer as to why. Treat them as best you can now, but when our therapy comes out, when the big pharma company brings it out, we'll have a solution for those patients.
Gotcha. Okay, that's great. Callie, there's a ton of audience questions, so I'm just going to let you take it away.
Okay, great. And Eugene, you answered a lot of them just in the last couple of minutes. So we'll get to some of the remaining ones. How does your precision medical approach improve the probability of clinical success compared to traditional autoimmune therapies?
Yeah, that's an excellent question. And again, I think at one level, it's by treating patients that we measure in advance are likely to respond well. So what we know is the traditional autoimmune therapies or immunosuppressives don't work in 30 to 50% of the population. So on average, the response rate is lower if you were to look at an overall clinical trial. What we're trying to do, as I described in a little bit of detail, is identify with our biomarkers in an outcome study before we run our clinical trial the definition of a population that's likely to respond to our therapy, but also likely to respond really well. Where if we normalize the protein, the benefit might be a 2 or 3-fold better response to a standard of care. So by defining the population most likely to benefit from our therapy with these biomarkers, we can totally change the outcomes as opposed to the normal approach.
And then how do the antibody, ASO, and diagnostic programs complement one another strategically, and which do you believe could create value the quickest?
Yeah, I think it's an excellent question. They complement each other strategically, I think, in a very important way. What I can imagine first with the 2 different therapeutic approaches. Remember, an ASO is essentially changing the recipe so that individual doesn't make as much of the protein, and the antibody gets rid of the excess protein that's there. Now, I could imagine those two therapies may be working together for some patients. Maybe there's some patient populations where one works better than the other. By having both, the large pharma company who acquires us, I think would have that opportunity to optimize therapy, but also they'd have the ability to block with IP someone trying to approach the problem we're trying to approach with, either an antibody or an ASO. Then the diagnostics are critical because of the precision medicine approach. Again, our view is the right thing to do in general in medicine is to treat a patient most likely to respond to your therapy. The more information you can get, and it's not always possible, but in our case, it's very possible to get exquisitely good information about what patients might respond best to our therapy. And that's what the diagnostics are for. Now, in terms of the value, I would expect the combination of these, partly for some of the reasons I mentioned. The big value equation is an M&A exit. Now, how do we build the strongest case? It's competition. But if we can make the case that our diagnostic will prep the market, so market penetration will be more rapid, physician uptake will be more rapid, and we have a broad set of indications, that all builds a very, very strong case for an M&A exit. But not to discount what I mentioned briefly with Stephen, which is that our diagnostic revenue could be an opportunity before we exit. I don't have expectations of it being too huge, but it could be a meaningful contributor to our economic, benefit, and, a value creator. Maybe it allows us to raise less capital with less dilution. We'll have to see how that plays out. But I think all three of them, can play a role, and we're working hard to make sure that we utilize the combination of all our assets to the maximum benefit.
Great. And what feedback have you received so far from pharmaceutical companies like J&J, Roche, regarding the biomarker and therapeutic platform?
Yeah, some strong interest. I think we're coming up with more data in rheumatoid arthritis. We partnered with a guy named Dr. Michael Weinblatt, who's at the Mass General Brigham. He has a very large data set of patients with RA and over 20 years of data. And we're going to look at things like correlations with response to therapy, correlations with severity. They'll really just beef up the case, incredibly well, that we're onto something. The biology makes a lot of sense. So many of the pharma companies we've been talking with are very eager to see that. Some of them have expressed some interest in thinking about utilizing our biomarkers for evaluating things in their own trials. That's still in the early days. We're going to try to keep those dialogue going. And part of what we're hoping is, as we invite pharma companies to maybe sponsor some of their key opinion leaders in our biomarker, biosample studies, that might get them thinking about, how do I use this in my own programs? I think we've heard also from people that precision medicine is very much in interest. You mentioned J&J. They've been very public, as has Amgen and a few others, about precision medicine as the approach that they believe in, that they want to take. So far, I think the feedback has been very, very positive, and we're going to try to keep generating good progress that keeps the discussion progression.
And so could an investor that focuses on medical devices invest in the diagnostics separately?
Not at this time, but we anticipate that will be the case going forward. So we've had some discussions at the board level about eventually spinning out a diagnostic subsidiary, which we then would expand with capital. And I talked, for example, about seeking an approval, like a PMA approval. We would definitely be interested in getting external capital to do that. And that subsidiary then might exit to a large diagnostics player like a Quest or LabCorp or Qiagen, and we've started some discussions with them. That investment opportunity would be down the road, but anybody who's interested, we could certainly keep them up to date on where we stand there. Either later this year or early next year, we might be in a position where we spin out that subsidiary and would be looking for investment capital.
And then you talked a little bit about your patent portfolio. What is the life of your portfolio?
Yeah, much of it was filed fairly recently. I think you probably know there's sort of, once it issues, you have a 20-year patent life. So I'd have to do the calculation but we're in the 20 years away from kind of time frame. So plenty of patent life, put it this way, so that we would be well positioned with a pharma acquirer looking at how much patent life would be left, when they were able to get to launch. And I think some of the approaches we're taking that I described of rapid accrual, streamlining, investigator interest in the trials, all of those shave time off the time frame to approval, and by virtue of doing that, could extend patent life. So I think we would be in the favorable zone of expected patent life at launch relative to what pharma is looking at in their other M&A opportunities.
Have you considered a non-dilutive deals on China, etc.?
Absolutely would consider regional rights deals, and China's a great example of an area where there's plenty of capital and it could be a very good fit. We're a bit early for that. Obviously, with those deals, you always want some sort of a buyback option so you don't preclude a big pharma transaction, but those can be done in a way that's very favorable, let's say, for the China rights groups. And yeah, China's a great example. One could also do a regional rights deal in Europe. But we're certainly, or Japan is another area where that can happen. We're very open to that. We don't have any active discussions yet. It's probably a bit early, but that could be a wonderful idea for a non-dilutive option.
Great. And then the one last question is, are you raising any capital now? How much, and any valuation comps?
Yeah, so we are actively beginning the discussion of raising capital by later this year. And specifically what we're looking at is a $6 million seed 2 round. We already have a little over 2.5 million or thereabouts spoken for interest from existing investors. So looking for an incremental call at 4 million. Now, the previous round was done as a 4.5 million pre. We don't have a lead term sheet yet, for this. So we don't have any specific pre-money value of that next raise, but expect it to be a modest step up from the 4.5 million pre in the last round. Again, looking for 6 million in the second half of this year.
Great. Well, that concludes the questions, and I want to thank the audience for all of the great questions that came in. If you want to dive deeper or want an introduction to the company, please reach out to me. Everyone has my email address. In turn, they will have all of your information as well. We'll be reaching out. And Eugene, I do want to turn it back over to you for any final words before we close out.
Well, first of all, just to echo, thank you so much for your time today. I really appreciate your interest in ABS. And as Steve started off at the beginning, I think there's a huge issue that needs to be addressed in autoimmune disease. 25 million people in the US have autoimmune disease, probably 10 million don't have adequate treatment. And that's a huge problem that needs to be solved. We think our scientists have discovered something that could be a solution for many of those patients. And we're really looking forward to the continued progress, hopefully showing that we're on the right track. And if any of you are interested in learning more, we'd love your support, and we'd love to talk with you more about, the possibility of collaborating with ABS.
Great. Thank you so much. Thank you, everyone, and have a great day.